So Ben, thanks so much for coming. I'll let you go into it. Many of us know you, but if you'd like to give a few comments about yourself, that would be helpful too. But thanks so much for coming.
Sure. So yeah, my name is Ben Domingue. I currently work at Stanford. Delighted to join you all today. Yeah, and as Jason noted, I have connections with just about everybody. So Jason was one of my kind of mentors as a postdoc. Sam worked with me while he was at Stanford. Lauren I've known for years. I was actually visiting Phil out in Europe when Donald Trump made his travel ban announcement, so I had to fly home on very short notice. And then James and I have had some drinks together at a few Madison watering holes. So yeah, it feels like a very nice group to be visiting with today. So I'm going to talk about distinguishing between interactions and dispersion effects in the analysis of G/E. And this is work I've been doing jointly with Clint Konopka and Travis Millard, as well as Sam, who you obviously know, and then Elliot at the bottom right has really been instrumental in this work. It's kind of, been a prime mover for all the things I'm going to talk about today. So, as you all know, we're kind of living through a moment where there's a real proliferation of genetic data, and in particular, there's a proliferation of polygenic scores. So many of the big data sets that social scientists have long used, like the AdHealth and the HRS and the WLS, are now releasing polygenic scores for those data. There are catalogs being made available that contain polygenic scores for an even broader swath of data sets. And I think one side effect of this is that as polygenic scores become more widely available, we're likely to start seeing a lot more gene by environment interaction research. Now, that's fine, but I think fundamentally G/E research is quite hard. And I think it's going to be even harder now that we have genes measured in population representative studies that contain environment with very large main effects. So when G/E was primarily being done in twin studies, oftentimes the environments under consideration there were environments with relatively small main effects, whereas the environment I'm going to talk about today is primarily going to be birth year, and that's an environment with a real big main effect that might have a number of it offers opportunities and challenges. And so the paper at the bottom right there was published recently, and this was kind of a first look at some of the challenges that we were thinking about in thinking about G/E research. And in particular, that paper started to describe what we call dimmers and prisms. And I have a kind of hand-drawn typology at the right. So a prism, right, the idea here is a kind of optical prism that inverts an image. And that is often the kind of conceptual model that people point to when they motivate their G/E research, right? The notion that a genotype might be advantageous in one environment and deleterious in another is something that has real kind of conceptual appeal, but it is, in my opinion, often not what we get when we're thinking about polygenic scores, especially. With polygenic scores, I worry that what we're often doing is actually studying dimmers. Now here, what I have in mind when I say a dimmer is kind of a light dimmer where you can turn down the amount of light or you can turn it up. And basically the concern is that the environment is really controlling variation in the phenotype. And that might have real implications for our G/E work. And the reason for that is basically heteroscedasticity. So heteroscedasticity is all about the error term in our models. Right, the error term is something we typically assume is very well behaved. We assume it's independent of everything. We assume it has a nice normal distribution. And there's just no reason that in general that need be the case, right? The distribution of the error term could be asymmetric. It could have a shape that depends on the predictors. And this heteroscedasticity, as we'll see, can lead to real confusion in G/E modeling. So I have a picture there on the right where we basically have, the dependent variables on the y-axis, the independent variables on the x-axis, and you can see that the variation in the error term, the variation in the dependent variable is dependent on the x value, right? And this is a challenge we have to deal with. And I can now replace that figure with one wherein we now have measured environment on the x-axis, some outcome on the y-axis, and then the three lines are showing slopes at three different levels of the genotype. And what you might kind of look at this and see is G/E, right? For I have a kind of changing slope across the environments that is potentially indicative of GxE, and yet that figure is generated entirely without an actual gene-environment interaction. That's all due to heteroscedasticity. So the scenario that I want to talk more about is as follows. We're going to be interested in a genetic predictor that predicts a constant proportion of outcome variance, right? So heritability or penetrance doesn't change in that sense. But the outcome is going to be such that its variance shifts as a function of the environment. And that heteroscedasticity is going to lead to GxE, and this is going to be something we try to address. And so we're going to get at this via a comparison of 3 models. Okay, so I'm going to talk through 3 models via a common set of figures. So let me tell you what the figure is. So in this figure, the x-axis is going to capture variation in environment. The y-axis is going to capture heritability, right? And so what we are basically asking is, how does heritability change as a function of environment? The classic notion of GxE is that that's what's going on. You have changing heritability as a function of environment. So the first model is just a kind of what I call the vanilla GxE model, right? This is just a classic gene environment model where you just have this interaction term B3 here, right? And this model is consistent with a scenario wherein we have changing heritability as a function of environment, right? You can see the kind of upward slope of these lines. They're not flat. All right, we're going to contrast that with what we call our scaling model, right? This model is shown here in an equation. I'm going to say a lot more about this equation on the next slides, so don't worry too much about that equation at present. But what I want to emphasize is that this model generates data that does not have a changing level of heritability as a function of environment. You can see that these lines are pretty much flat. And if you think about this expansion, we'll see this specifically in the next slide, but you do get a GxE term here, right? So there's GxE in the sense that you do have the kind of product of G and E, but it's a very delicate kind of GxE that does not lead to changing heritability across environments. The third model is our kind of generalized model that captures both models 1 and 2. It's a model that basically contains everything we care about. It contains main effects for gene and environment, a GxE term, and then it also is going to allow for environmental heteroscedasticity, right? That's the key part. So let me kind of talk more about models 2 and 3. Oh yeah, so the goal here is going to be to estimate model 3 and then to ask whether model 2 is a sufficient reduced form model for describing the data. Okay, so I want to make a few points about model 2, the scaling model. So that model is motivated by what I'm showing here. In particular, note that Y star bit. The punchline is that we control the heritability and we keep it constant based on Y star, right? So Y star is the part of the phenotype that's affected by genotype. And you can see, just given the way we've written it down, the effect of the genotype is constant for Y star. What's varying is the variation in the phenotype, and that's driven by this term here. Okay. If we expand, this kind of thing, if we multiply, if I replace Y star with this thing over here, and then I multiply through, I do get a GxE term, right? That's right here. But what you don't really see just from this kind of view is that there's actually a kind of delicate balance of the covariates in this. In particular, there's a delicate balance of the coefficients that we're now going to exploit. Because what you can do is you can equate models 2 and 3 and ask, when does model 3, right, this one up here, reduce to model 2? And what you find is it's precisely when this equality holds, right? When pi 1 lambda naught equals pi naught lambda 1, right? When that holds, model 3 reduces to model 2, but of course this doesn't generally hold, right? This, it just need not hold. And so model 2 is a special case of model 3, as is model 1, and what we're going to be interested in primarily is asking, when can we basically use model 2 as a reduced form of model 3? And that's going to really amount to testing this test statistic that is, exactly what I showed you here, right? I've just taken this, subtracted everything to one side, and I basically am going to ask whether this test statistic, which we call GxE, whether this thing is zero, right? So if this test statistic is zero, then model 3 reduces to model 2. And in that case, we have the kind of special case of G by E wherein heritability is actually constant. Remember the figure I showed you a few slides ago. In the paper, what we really get into is how to estimate this statistic via maximum likelihood. And if the thing works effectively, we can really adjudicate between models 1 and 2. We can ask whether, estimated G by E in the sense of, kind of a coefficient on the interaction term, whether that's due to kind of the G by E we have traditionally cared about that's associated with changing heritability across environment, or this weird form of G by E that's just due to kind of scale oddity and variation that's going on. So does this work, right? Can we actually use this test statistic reliably? There are a lot of simulation studies in the paper, so I'm just going to quickly summarize these here. At the end of the day, this test statistic is like we're just relying on very conventional maximum likelihood theory, so there's really no reason to worry too much about it. If we're working under the null, where model 2 is true, and we estimate the test statistic using estimates of model 3, right? So we estimate model 3, plug those estimates in to this form for the test statistic, and then consider its shape. It ends up being a chi-squared distribution, which is exactly what we would predict, right? So the red curve here is just a chi-squared statistic for the appropriate degrees of freedom. The blue are that this is a density over a bunch of simulated data, and you can see these two things line up perfectly. So the test statistic behaves perfectly well under the null, but what about if the model is misspecified? What about the case wherein we have something else going on? What happens? Well, what if the true model is actually the kind of classic G by E case, and we are asking about whether the scaling model actually produced the data? Well, the punchline is everything works just fine. If we have relatively large data sets of several thousand respondents, we're very well powered to reject the null that XE equals 0 and that the scaling model is kind of sufficient to capture what's going on in the data, right? We would reject the scaling model and prefer the vanilla G by E model. If we have fewer data, right, if we have only about 1,000 and we still have a decently sized interaction coefficient, we can also perfectly well reject the null that the scaling model generated the data. So if we have vanilla G by E, we can use XE to kind of rule out that heteroscedasticity produced it, which is, I think, quite valuable. What about a different case? What about if we have heteroscedasticity, but no G by E at all, right? So this is a case that, we have kind of, a shifting amount of variation that's controlled by the environment and the outcome, but no G by E. And the punchline is we can kind of put information together about the G by E term in the model and the XE statistic and kind of make sense of this. So here, what I'm flagging is basically that we can detect that the pi1 coefficient, that's the coefficient on the G by E interaction, is actually 0, and then we don't have to worry too much about XE. XE gets more complicated here, but if there's no G by E kind of signal to begin with, we don't have to worry about whether it's due to scaling or not, right? Here, there's just no evidence for G by E, and we then don't worry about the fact that we don't worry about XE because it's only relevant if there's actually G by E to worry about in the first place. So let me pause. I don't know how you guys normally run these. Are there any questions? I'd love to, especially if there are misunderstandings or point of confusion, maybe we can talk through those.
Hi, can you hear me?
Mm-hmm.
Hi, my name's Ava. I'm a graduate student here. So I'm actually having a little bit of trouble following along. I can't, I guess I have trouble following along looking at models on the fly. And so I was hoping if you could just go back and, or at least confirm if I've misunderstood something. So is the main concern that when we have this different variation across an outcome, that what we observe as a G by E interaction may actually not be a G by E interaction, that it's really just due to the variation or to the differences in heritability across the particular outcome that we're looking at. And so, and then what I haven't, I guess maybe what I don't understand is how this statistic relates to identifying whether or not that's happening in our models, and how, because the statistic is derived based on the intercept and the slope, and I don't know how that represents, yeah, I got a little lost there.
So yeah, let me take a stab at explaining this again, because I think it is, some of these things are relatively subtle. So going through it a second time is worth it. Our main kind of concern is that we have traditionally cared about G by E in scenarios, well, for reasons like this, where we have changing levels of heritability as a function of the environment, right? You know, basically we have low heritability at one environmental level, high heritability at another. What we are arguing is that there's an alternative scenario where heritability is constant, but you can have kind of a form of G by E that's introduced that is not actually consistent with changing levels of heritability. It's just consistent with changing variation in the outcome that's environmentally controlled, right? Hence the dimmer notion. If I turn the dimmer way down, there's not that much variation in the environment. Genes aren't going to explain that much variation because there's not much variation to explain. If I turn the dimmer up, there's a lot more variation. The genes can explain a lot more in the absolute sense, but relatively speaking, we're always explaining a kind of constant level of the outcome variable. But we can be easily confused by changing variation in the outcome. So that's the kind of conceptual motivation. And what we then do is basically kind of write down what is effectively the dimmer model, right? That's this one here. And then I think there was some confusion about the test statistic. The test statistic is basically based on these, and this is a combination of pi's and lambdas. The pi's are the main effect in the interaction term for G. The lambdas are the kind of just the pure residual variance, and then the environmentally controlled residual variance. And if these things kind of form this delicate ratio, that's when we get our scaling model, right? And so basically what we're going to do is put information about all these parameters together to construct our test statistic. If that test statistic is 0, what we're saying is basically that is evidence to suggest that this model, right, number 2, is sufficient to explain your data. And in this model, you don't have variation in heritability across environment. You have constant heritability across environment, but changing levels of outcome variation. Does that help kind of explain conceptually what's going on?
Yes, that clarifies things. And then, so to get this, so you're trying to explain to us how we would be able to get this statistic in order to tell if we have, if the dimmer model applies. Is that, okay, that's where you're going next.
Right. Yeah. So I'm going to show you some empirical stuff where I hopefully walk you through a use case. And then we also have kind of provided some software that you could use to actually estimate this if that's what you were hoping to do in practice.
Ben, could I draw out one other question? It's related to Ava, which is maybe it's the opposite, which is to say that heteroscedasticity is probably just a general annoyance in any interaction models. I'm wondering if the box number 2 is meant to be at all specific to G by E versus a context where there's no genetics. Is there anything very specific to G by E that you're wanting to highlight?
No, you're absolutely right. This is a generic kind of thing with respect to heteroscedasticity. So, you know, we've talked to a bunch of people and so one possibility is that somebody has made this observation previously in another setting. We don't know if that's the case. We haven't seen it. We've talked to people from a variety of disciplines and nobody's kind of pointed us to a precursor of this basic idea. So absent us missing something, we think this is novel, but it's also true that it's just generally applicable to scenarios wherein you're studying interactions. So the empirical context that I'm going to show you in a minute is going to be, I think, hopefully, illustrative of how you could use it in GxE settings. And I would also note that it's coming shortly, but one other thing that I'm going to say is that you can also do work in the kind of VQTL space with this basic model. And so that's another reason I think that kind of GxE work and genetics work in general might be interested in this basic approach.
Thanks.
Can I have a question?
Okay.
Can you say a little bit more about, so, I mean, in some sense, it seems like there's different ways to define GxE, right? Like when we were in the twin study world, necessarily they were just testing like whether the heritability varied across environments, like the kind of classic like SES, for example. But now that we have molecular genetic data and we're able to do like classic regression modeling, someone might say kind of like that beta 3, sort of this mean difference across environments is what we care about. Like that's actually GxE. It's not heritability changing. It's this first moment thing. I mean, what would you say to them, or like why is it important to distinguish between these two things?
Oh, I mean, I think it's fine. I'm not here to tell you what you should care about. I just think we should think clearly about it, right? Like we should understand which of these models is giving rise to the thing we're observing, because they have very different substantive implications.
Right, and so maybe that's my question is like, is there a reason why, like say that you use the vanilla GxE model and you get a significant beta coefficient from like beta 3 that says there's GxE, but it's, in reality, if you were to fit these three models you're presenting, you would learn it's just like all driven by heteroscedasticity. Like how does that change how people you think it's like reasonable for people to interpret that coefficient?
Yeah, well, I mean, I think it's fine to, like, I think maybe it helps to talk about this in the empirical context that I'm going to introduce briefly, but, I think if you're substantively interested in BMI, I think it makes sense to ask about the role of genes. I think it makes sense to ask about the kind of role of environmental exposures, but I don't think it helps to get confused when you put the two together and then mislead yourself about what's going on. But yeah, I mean, I think my main point is just that you can still be interested in the fact that the absolute kind of magnitude of the GxE coefficient is increasing, even if heritability is flat for substantive reasons. That's totally fine. I just think you want to know that that's what's going on.
Yeah, and I think part of the reason we're motivated to do this, right? I was just trying to get at the idea that like there's like really stylized interpretations of like people will just run a basic regression model with interaction, and then like social scientists love this idea that like genes are contingent on environments or environments are contingent on genes, and then there'll be like sort of really big extrapolation away from like, ooh, like this is probably going through this process or that process, or like when, in some sense, maybe the simplest answer is that like, anything you interacted with that had a main effect would have shown this, or, it's heteroscedasticity.
Yeah, I mean, Sam, like, I think you raise a nice point. I think maybe the reason I'm interested in this will become a little clearer when I talk about BMI and birth year, so maybe we can revisit at that stage.
Cool, sounds good.
Any other questions at this stage? So if not, let me note one other thing we can do with our basic model, and that is we can study VQTLs, or the variance quantitative trait loci. So, traditionally, approaches like GWAS have been interested in looking at those loci that affect the mean of some phenotype. More recently, there's been interest in looking at those loci that affect the variation in some phenotype. There is, as I understand it, a kind of biological reason that this might occur, right? There's this idea of evolutionary capacitance wherein the genome is basically primed to unleash variation in the world when needed in the context of some novel environment that the kind of existing genetic status does not kind of appropriately prime for. So, this is the kind of thing that people are starting to work out. There's this recent paper from the Vischer lab that talks about this kind of thing. I stole one of their figures. And the punchline is basically that we can extend our model in a very straightforward way to ask about genetic variation that's associated with heteroscedasticity in the outcome. In particular, We just replace what we used to have here, which was a term that involved a product of the environment in the error, with now a term that is the product of the genotype in the error, right? And so this is going to be a very straightforward kind of switch, because in the equation, E and G are effectively symmetric, and so it's, you know, all the math works the same if I replace kind of lambda times EI with lambda times GI. And I'll show you some results from this kind of approach, but I just want to flag that, you know, this is an active area. So there's this paper from Rebecca and Dalton looking at polygenic scores for plasticity. I think somebody from Wisconsin used it to do some work that was presented at IGSS. I think there will be a lot of work in this space in the near future, and so I'm emphasizing that you can use our approach to kind of use such scores potentially in more refined models. Okay, so let me now turn to the empirical example that will hopefully both kind of illustrate why I care about this and also illuminate some of the inner workings of how we're using it. So, as you all know, at this point, there's been a lot of work on the genetics of BMI. In particular, there have been a number of very high-quality GWAS of BMI. And people have started to use the results of those GWAS to examine shifting patterns of genetic penetrance as a function of birth cohort. So the main paper that I have in mind is this paper from a few years back that was published in JAMA. And they basically observed the polygenic score for BMI is more penetrant or increasingly predictive in more recent birth cohorts. So these birth cohorts, they're basically HRS birth cohorts. So the green line represents the slope associating the PGS for BMI and actual BMI. This green is the earliest births. Yellow is 1924 to 1933, and so on, right? And so you get to the top, and now we have the steepest association between the BMI PGS and observed BMI, and they're taking this as evidence for GxE. I should note that other folks have made a similar observation. So Hechuan Liu and Guang made an observation that was published in ASR. I did some work on this with Dalton as well. And so this is something I've been interested in, and what I'm going to present from here on is relatively critical, but this is not meant as an attack. So I would be implicated in that attack as well. So all the data that, well, these three studies used was from HRS, so that's what I'm going to use here as well. And the problem is that there's been a very important fact about BMI that was really not accounted for in these prior papers. So I'm showing you chickens here. If you didn't know, the kind of body size of the average broiler chicken has just exploded in the last half century. And we, in fact, have the same basic problem with humans. I pulled a table from the International Journal of Obesity that suggests for respondents that are 50 plus, right, that are roughly the same age as the HRS respondent, there's been something like a 50% increase in class 1 obesity between 1960 and 1994. This is a huge shift. You can observe this in the HRS data. What I'm showing you here are BMI quantiles for what are roughly 64-year-old respondents at wave 12 versus 64-year-old respondents for wave 2. And you can basically just see that the quantiles for the wave 12 people are just a good bit higher than the quantiles for the wave 2 people, right? This is consistent with this notion that BMI has increased and variation has in fact exploded as well. Here's another picture, right? I'm showing you now birth years on the x-axis and BMI on the y-axis. And I have, I believe, the 10th and 90th quantiles sketched out as red lines. And you can see a kind of an increase in the 90th percentile over time, right? We have an expansion in the amount of variation in BMI as a function of birth year, along with a kind of upward shift in the mean. And so the concern would be that this heteroscedasticity, right, this increase in variation in BMI as a function of birth year, may be an issue that needs to be resolved in our analysis of this BMI by birth year interaction. So here are some results, right? And so these are, basically conducting the same analysis that was done in earlier work. We have results here shown for both genders, then separately for males and females. And we also use the kind of raw BMI metric and then a transformed metric that is meant to remove some of the skew in BMI. I can say a little bit more about that later. But I want to talk through what these estimates are telling us. So these are all estimates when we estimate model 3 from the earlier slide, right? That's what I'm showing you here, right? We have the pi's and the lambdas. So what do we have? Well, I want to start with the males. So the first thing to note is that we get kind of very significant G by E coefficients, right? The pi 1 term, you know, you would kind of, especially for the raw BMI metric, you would you know, reject the null that pi 1 is 0 for any conventional level of alpha. But we have very pronounced heteroscedasticity. I'm not even showing p-values for these terms because they're under 1 × 10⁻⁶ uniformly. You know, we have real heteroscedasticity problems here, right? So we have, you know, kind of big pi 1 and big lambda 1. And so now there's a question about, well, do these two things balance in the way to make that test statistic C equal to 0? And for the males, the answer is basically that they do. So for the males, we fail to reject the null that our scaling model is sufficient to generate the data, right? So these are p-values of 0.39, 0.38. We don't reject the null that C equals 0. That short form, the reduced form model 2 seems sufficient to explain what's going on. And I can show you what that looks like in a little more detail. So what is this going to imply for our understanding? Well, to illustrate this, I'm going to go back to these slides that I showed you earlier and use what's, yes.
Could I ask you a question? This is Alberto Palloni.
Of course.
You've written a paper on the effects of selection. Stefan did not adjust for selection at all. I wonder whether some of these results could be affected by that, since part of the selection, what it does, it reduces the variance as well as propping down the BMI.
Yeah, that's a great question. I think I generally agree with your intuition that that might play a role. So that might be another reason to be concerned with those results. I'm going to talk in a bit about the UK Biobank briefly, and I'll note that we have a result there that is kind of, it's very strange unless it's all due to selection. So I would generally agree with you that selection is a very big problem that is under scrutinized, although I do think folks at Wisconsin, in fact, are looking at this with respect to the UK Biobank. So I agree with that observation that that's a really important phenomenon that I'm not talking about.
Thank you.
I think I'm restating Alberto's concern a different way, which is that your models assume that gene-environment correlation is 0. Is that a maintained assumption, or is that allowed in some case, in some kind of generic case?
No, we don't make any requirement about that at all, I don't think. I mean, at some point they could be sufficiently collinear that you might start to have estimation problems, but we don't make any assumption about G&E being independent.
Is that to say that you think that there's no requirement for genes selecting environments? So like Alberto's describing genes selecting environments in the case of actual people not being in the sample because they didn't survive long enough to be in genotyped in 2006 or whatever. But just a larger class of people, that genetics being informative about which environments you're in is, I'd be surprised that the answer is that it, like, any level would be fine. And I wouldn't be surprised if the answer was that it's just a maintained assumptions that RGE is equal to 0, but it seems like it's somewhere in between.
Yeah. So I'm thinking through the simulation work we did. That's an interesting observation that maybe I should not say anything too strong about, because I don't think we built correlations between G and E into our simulations in a highly strategic way, so maybe I can revisit that. To the extent that you're making the point that that would be an alternative reason that the observation from that JAMA paper could have occurred in a kind of spurious sense, I would agree with that.
I guess what I was trying to get a sense of is whether, I'm restating my question a little bit, which is to say that, is this model best thought of to sort out heteroscedasticity versus G by E after you've done a lot of house cleaning in terms of RGE? Or is it like a top level question that could subsume RGE questions? Because besides the selection issue that Alberto raised and that you already know about based on your own work in the HRS, you might just assume that there is RGE between birth year as environment and genetics, as you've demonstrated. But whether it's consequential for your model, that's where I'm not sure if I should think of your model being the best case being where you've already cleaned up RGE and don't think that it's a major problem, or not. I guess maybe that's something to come back to a little later.
So yeah, let me continue to mull that over. What I think I'm confident in saying now is that the levels of association you would get, I'm pretty sure, between birth year and the BMI polygenic score, they're probably significant given the sample size of the HRS, but I think they're low enough that I don't think they're likely to really distort too much what I'm about to say.
Thanks.
You buy that, Jason? Okay. So the way I'm going to now kind of unpack the results from our analysis is in terms of basically posterior predictive checks. So what we're going to do, we're going to take these gray lines that I showed you earlier, right? And these are all coming from simulated data, where I basically simulate data using parameters derived from the HRS. I simulate data first under the vanilla G by E model and then under the scaling model. And then I'm going to fit the full model, model 3, and show you the same Y quantity. All right, I'm going to show you the same thing on the Y-axis, now from the empirical data. And the question is just going to be, does this new line look like the lines given to us from the model under simulated conditions? And here's what we see, right? So the red is now the result from empirical, the actual HRS data for the males when I estimate model 3, right, the full model. And what you basically notice is that there is not much actual change in the amount of outcome variation explained by the genotype for the empirical HRS data. Way less change than you get from the vanilla G by E model. The level of change is in fact quite consistent with our scaling model, right? Which is precisely what we learned when we were unable to reject that XE statistic is 0, right? That's equivalent to saying that the scaling model is a sufficient reduced form approximation of model 3 to generate our data. This is for the males. We can now talk about the females. And the story is a little messier. So for the females, you see that there's a little more change. Well, let me start over here. You have more change in the amount of outcome variation explained by the genotype over the birth years under the empirical, in the empirical data than you do from the data simulated via the scaling model. But it's also too flat to really look like it comes from the vanilla model, right? So the data are not consistent with either reduced form model, model 1 or model 2. And what we can do is we can decompose elements of that pi1 coefficient from model 3 into scaling and vanilla components, and basically assert that roughly 60% of the G by E that we observe here is due to scaling, right? For the males, this was a bit higher. It was 75%, and that confidence interval included 100%. And basically, this allows us to say, that the lion's share of what we're observing in terms of G by E is really just due to the scaling problem. If we now look at the full data set, we can kind of play the same game with the decomposition and observe that 63% of the variation in the in the G by E that we're estimating is due to scaling. So my best guess at this stage, if you ignore the kind of problems of RGE and selection, if you just kind of take the data, on the ground in which it's presented in that original paper, my best guess is that their finding is largely driven by changing variation in BMI across birth year. Questions about that before I move on to the next example?
Ben, just
Yep.
just another question. Most people would like, well, most conclusions are derived from significance tests, and so people normally look at the number of stars you have in the regression curve. The question that I have is, have you done the exercise of eliminating the part, eliminating the part that is due to the scaling and trying to see whether the coefficients are still significant? Namely, it would not prevent a person from insisting that there is a gene environment interaction.
Yeah, that's a good question. We have not. For the males, I feel pretty confident asserting that there would not be, given that the scaling model is statistically a sufficient approximation. For the females, I'm not entirely sure. That's a good question that I don't have an answer for at present.
And that jives relatively well with the fact that the BMI increases are a lot stronger among women in general, and I presume in the US as well, than for males. And so there is a lot, well,
yeah, my guess is that for the females, you would still get some significant kind of G by E. Now, so two things about that. So first, I think with this entire paper, it would just be much more informative if it had really been clear about the models that were kind of giving rise to the data, rather than just noting this this increasing slope over the birth cohorts, which is, in my opinion, just it's not the right way to kind of mentally model what's going on. As I admitted earlier, I had made the same mistake, so this is not to be overly critical of them. The second thing I would note is just that even if that's the case with the females, that we can't forget that might have to do with any number of other problems, selection being one. Also, the nature of the BMI GWAS might be such that it's more predictive in more recent cohorts, just as a function of the way the GWAS was put together. That's something Dalton has been interested in. So yeah, there are many mechanisms that could give rise to that basic pattern.
Good, thank you.
Yeah.
I have a question, Ben, and so I see what you're saying, and I understand that, yeah, this kind of changing variation in BMI over time is kind of what's mechanistically kind of moving this, or showing this G by E result. And I guess, yeah, and maybe this gets also to Jason and Alberto's question about, RGE. I mean, I guess in the sense that, part of the reason that the distribution might be different now is because these variants that, are kind of BMI enhancing have been allowed to have more penetrance over time because we're in a more obesogenic environment. And so it could be that the genes are part of what's shifting the distribution. And so, yeah, I guess I'm having trouble kind of defining that as kind of pure heteroscedasticity in my head, or I don't know, I'm having trouble kind of thinking about how that's not.
Yeah, so that's a fair point. I mean, I think at the end of the day, what I would say is, especially for males, we should be satisfied at this point with a mental model wherein what's happened is that there's been an explosion in the variation of BMI as a function environment that has nothing to do with the genes. And the increasing penetrance of the genes is just along for the ride because there's more variation to explain. And once you correct for that, the genes are kind of irrelevant in that sense. The story for females might be more complicated. And I mean, your basic notion that there could be, well, you're effectively asking now about the VQTL part. So let me turn to that because I think that's what's kind of coming next. And that's, I think, another way of interpreting your question. That's an interesting one. And we have a little bit to say about that.
Yeah. I guess I was going to say that too, because we're doing that VQTL study and with BMI, and we can talk about this more in our meeting too, but yeah, to what extent then the VQTL PGS is kind of controlling for some of that kind of heterogeneity and whether that's kind of capturing the same thing that you're saying. Yeah.
So let me talk about those results. So I've largely talked about polygenic scores up till now, but there's nothing in the basic setup of our model that restricts us to polygenic scores. You can play the same exact game with, you know, kind of allele counts at a single locus. And so we're actually going to do that. And so we're going to do that using the UK Biobank. In particular, we're going to look at the 100 top hits from the LOC 2015 BMI GWAS. That was, I believe, the last BMI GWAS to not have the UK Biobank in it, so that's why we're looking at those 100 top hits. For the purposes of the talk, I'm going to talk about the VQTL part. In the paper, we do the kind of MQTL bit as well, but here I'll just talk about the VQTL bit, and recall that we basically ask now about variation in the outcome that is associated with genetic variation, not environmental variation, which is what I was talking about before with the HRS and birth year. So here's the relevant table. So we basically had 96 SNPs out of the 100 that we could find in the UK Biobank. These are the 6 that were flagged as VQTL SNPs in our analysis based on an analysis of what we call lambda 2, which is just the genetically linked heteroscedasticity component. These all have, you know, highly, after we adjust for the multiple testing, these all have significant amounts of genetically linked heteroscedasticity. And there's one in particular that's interesting. That's this guy right here, right? This is a SNP that's in the FTO gene. And we basically, you know, we reject the null of the scaling model. We reject the null of the scaling model and end up with a scenario that is very consistent with earlier work suggesting that FTO may be associated with both birth cohort variation in BMI, which is the MQTL part, but then also phenotypic variability, this plasticity part. That's this paper from 2012 in Nature, right? So our findings kind of are consonant with those in the sense that some of the loci in FTO might be playing relatively confusing roles wherein they both have changing effects over time and then also may be associated with changing levels of phenotypic variation. Now, this is the thing I wanted to note that is going back to Alberto's earlier point. When we look at the main effect of birth year in the UK Biobank data, we get a negative coefficient suggesting that later born respondents are predicted to have lower BMI. That is highly inconsistent with a lot of epidemiological evidence, right? And I don't know what this is due to exactly. My guess is that it's due to selection issues. So this, I think, would be, you know, this is a potentially fruitful ground for follow-up. But we do observe increasing variation by birth year, right? So our basic heteroscedasticity notion remains relevant in the UK Biobank as well.
One quick question on that, Ben.
Yeah.
It seems like for the UKB, you have an age period cohort issue that you don't have as directly in the HRS, where that knowing someone's birth year in the UKB, you know quite precisely what their age is at assessment. Whereas in the HRS, you have some room to play with what age you're assessing the BMI.
Yeah, that's a great point. I mean, that's a great point that wasn't really that kind of sufficiently addressed in the JAMA paper or our paper as well. So, I mean, I think that's been a kind of underlying problem throughout. It's potentially it's something that will be harder to address in the UK Biobank, but it's something that one should be concerned with in any event. And so I think that on the next slide, I have more to say about BMI. When we measure it is something that's probably been under considered, and also the precise nature of that distribution is another thing that has not been sufficiently considered.
But this particular main effect of birth year, you could call it a main effect of age, and you would be approximately correct.
Yeah.
And so that you've switched the environment sufficiently. Like you said with HRS, you could conduct HRS with only measurements around age 65 or something like that, and get most of your birth years, so that you'd actually rule out age as the cause, except for this backdoor issue of selection into the sample who are genotyped. But here, like I said, you could just call this an age effect rather than a birth year effect, and you couldn't tell the difference, right?
Yeah, I agree with that. That's a fair point. So with that, let me start to make some closing remarks. So the first thing that I want to talk about is, in some ways, kind of the big question: is this scaling G by E that we've observed, is it actually G by E? I don't know that that's a really useful thing to argue about. To me, our approach is meant to offer additional insight into the processes that gave rise to data. This is not meant to negate the salience of earlier findings. It's meant to provide additional insight and intuition about what was actually going on in the world. So I don't particularly care whether you want to call it G by E or not. To me, it's more important to understand where the thing came from, or at least what models could have potentially generated the thing. So I described earlier this idea of a dimmer. I think that's like a stylized form of G by E that we should think more about. And the basic notion is just that E might in fact control a lot of the variation in outcome, which could induce G by E. And for conceptual reasons, I think this is likely to be a bigger problem now than it was historically. As I mentioned earlier, historic study of twins that involved G by E often involved environments that didn't necessarily have huge main effects. But now in population scale data, we have environments with massive main effects. Those main effects could be such that they introduce kind of additional problems, such as the one we've eliminated here, when it comes to study of interaction with those main effects. In the case of BMI, there are additional reasons to be worried about the distribution. So BMI has, a relatively nice distribution as far as these things go, but it does have some skew. That skew could be a bit of a problem. It's something we tried to address in our paper via some transformations, just to make sure that it was not the thing driving our results. But I just want to flag that I think, outside of height, most of our phenotypes have psychometric properties that lead to issues when doing G by E work. As was noted earlier, some of most of what I've said, the basic models aren't really about G by E per se. They're about interactions, and a lot of the psychometric properties of our outcome variables cause real chaos if you want to study interactions. So if you have a floor in your outcome variable, for example, that really just destroys both false power or false discovery. It really elevates the level of false discovery when you don't in fact have the effect, and it really distorts power if you do. So I just want to flag that measurement properties of the outcome variables are something that we haven't thought much about, but we really need to. And finally, there is code available at that website. I think you should all have the slides. You could also just Google my name on GitHub. And the goal here is to really help people consider these kinds of scaling analyses alongside more vanilla analyses that have long been conducted. By making this code available, the hope is that people actually can do that. I'm happy to talk more about using that software. It's definitely in, what's pre-alpha level of development. It'll work, but it's a little ugly. So I'm happy to talk to anybody that wants to try to use it. With that, I will kind of stop with the top part. I'd love to answer any questions. There is a paper posted to the bioRxiv. I'm going to update it, including in the updated title in the next day or two, so that just might be something to be cognizant of. Please feel free to email me if you have any questions. And again, thank you all so much for having me. This has been a real great opportunity.
Hey, Ben, it looks like Yushang has a question. I'm just going to read it because his mic's not working. So suppose the true model is the scaling model. However, when we do the test, we put the wrong E in the model. What will happen? Will we reject the null and believe that the presence of VQTL effect? Or should we interpret the test results such that if we do not reject the null, then probably there's no VQTL? Whereas if we reject the null, it's still possible false positive. Thanks.
Well, so let me make sure I understand. So the issue is that you have the correct environment is associated. The scaling model is the right model if you have the right environment. You, in fact, measure some correlated environment. And the question is, what does that do to resulting inference?
I think so.
Okay. That's an interesting question. You know, my gut is that if it would really just depend on the level of correlation between the two environments. So, you know, if you're measuring, so let me talk about this with respect to reliability. If you're measuring the environment you care about with the reliability above, say, 0.9, I don't think you're going to have major problems. If it gets low, things could definitely be concerning. In the sociological science paper that I referenced at the beginning, we do some power analyses showing the effect on power of lower levels of reliability in your measure of the environment. So I think that's an interesting concern in general. I don't have a perfect answer for how it plays out here. So I like that question, and maybe I would suggest looking at that science paper for at least one look at how noise in your environmental measure can distort G/E analyses. That's a nice question.
Hi, this is Q. I have two questions. First is just a point of clarification. So you said that this heteroscedasticity model can be potentially applied to identify VQTL. I wonder, in that case, do you actually need to know what the environmental factor is?
Yeah, there we don't use the environment at all. It's just purely the genes going into the model.
I see. So it behaves similarly to, for example, Jian Yang or Alex Young's work. So it's really just, you don't have to know the E, right? So across different genotype groups, the model can actually detect the variability change.
That's right, yeah.
I see. So the second question is, I've been trying to construct a hypothetical example in my head to explain some of the theoretic models you built. But so this may be an oversimplified example that's too specific, but suppose you have different birth cohorts, but then in one birth cohort, you know, fast food was not invented, and in another, you know, there's a lot of fast food. And then basically the genetics underlying BMI can have two different pathways, right? One is just general metabolic traits. Another is related to how much you are addicted to fast food, right? But these are very different genes in the genome. So then in the second birth cohort where you have this environmental exposure of fast food, a different set of genes are turned on. Now these genes actually have effect controlling the variability of BMI, but turns out it's just out of the additional amount of variability of BMI, these genes contribute a similar proportion compared to the first set of genes. Do you not interpret this as G/E interaction? Because if you look at individual genes or genetic variant, their effect do seem very differently between two different cohorts. Just when you aggregate the effect together, when you talk about heritability, the proportion seems consistent.
So that's an interesting question. So I don't think I disagree with you that would be G/E if it were as you described. My point would be that that's not the evidence we've been given thus far. So like if you broke down the JAMA paper used a top hit score. I think it was the 32 SNP score from the original big BMI GWAS. If you broke that down into SNPs associated with metabolic function and then SNPs associated with something else and showed like very different patterns across those two groups, that would be interesting. It wouldn't necessarily get you around the concerns I've raised, but I'm not disputing that as a G/E observation. My point would just be that that's not the data we've been given thus far. That, like, the evidence doesn't suggest, you know, that would be a kind of an additional assumption we would be making about what's going on, that it's maybe consistent with the data, but certainly not a necessary and sufficient explanation for the data.
Yeah, I agree that this may not necessarily be the truth, but I'm wondering, suppose this is true, would your model give you the null result? Would you be able to tell there's some G/E interaction going on, or would you come to the conclusion that heritability remains consistent? Therefore, we interpret this as there's no interaction.
Your claim would be that your model would be that basically I have a score that's constructed of two classes of SNPs. And in one environment, one class predicts. In the second environment, the other class predicts. But across the environments, prediction is basically flat because what genes are predicting has changed, but the overall level of genetic prediction has remained the same. Is that?
Yes. So you can imagine the first set of SNPs have constant effects in two cohorts. Another set have zero effect in first cohort, but the positive effect in the second. But then you have this non-measured, environmental factor that also kicks in the second cohort. So the overall heritability doesn't change into both cohorts.
Wait, why does the overall heritability not change in the second cohort? I mean, once you have
because you also have the environmental factor that has a zero impact in the first cohort, but a non-zero effect in the second.
Oh, you have the introduction of both a new class of SNPs that are predicting variation and then also an environment that's doing something.
Yes, but just the ratio of contribution between genetics and environment remains the same.
I guess I'd have to think more about that. I mean, that seems like a pretty slick counterexample that yeah, might, under our kind of, it might be scaling G by E in our typology. I can understand why one might not be satisfied with that, but
I think I have some stuff to add about this, because Ben, this is what we were talking about with Elliot like the other day, basically. And I think you and me are both sort of fine with, like, maybe over rejecting that and still calling it not G by E, where Elliot's like, well, this, like, he's saying the Q point, which is like fundamentally these are sort of conceptually distinct pathways, even though, so we should think about it as G by E. But I mean, I guess, like, Q, to your point though, yeah, I mean, you're right, I think, in that the only thing that we can fundamentally test, right, is like we have a sort of, genetic predictor, a moderator, and then the error term, which is like just everything else. And we're just asking, basically, does the moderation of the genetic predictor sort of outpace the moderation, or underpace even, the moderation of the error term? So, like, to the extent that there's, a bunch of different things inside of those things, and those are changing in a way that, like, is proportional, then we'll miss that. But yeah, I mean, this is sort of to Lauren's point too, which is that, like, I mean, so, I guess one way to think about it would be like, if the heritability of a trait is 1, then there's no distinction between heteroscedasticity and G by E, right? Heteroscedasticity is G by E. Like, the way that we're defining heteroscedasticity is sort of the extent to which non-genetic stuff in our model changes across environments. But sort of that also, or basically we're saying heteroscedasticity is what everything, like the variance of everything changing across environments, but we can only identify that using the non-genetic stuff because we also have this G by E term in there, which is like capturing sort of the classic G by E. So I guess Lauren was saying, like, well, don't genes sort of play a role? And I think they totally do, like, in these changes, from 1920 to 1960 and average BMI, birth cohorts of average BMI. And I think the question, like, we can't really, I mean, they're always going to play a role, right? Because, like, humans are just native genes and it's such a big part of our lives. But I think the thing that we're able to test basically is, like, do genes seem to play any amount, like, a different role than sort of everything else? And that's the question that we're kind of asking.
I would also add to that, like, I mean, so you raise a really interesting kind of question, Q, and so I would just say that, fundamentally, we're not hoping to incorrectly classify relatively kind of elegant models. That's really not the aim. This really kind of grew out of my frustration with being asked to review a bunch of G by E papers that are just doing very simple things and kind of drawing very big inferences that are quite likely wrong. And so the hope here, I think, is really just to get people to move to a class of slightly more complex models that might elucidate some of the features that are really driving what's going on. But again, if you have some really sophisticated model for how things are working, this approach might not be sufficient to capture that kind of full complexity.
Yeah, I see. Yeah, this is very helpful. Yeah.
This is James. First of all, I want to say thank you for the really great presentation. And I've had like a few minutes to process and reflect now. So listening to some of the questions that have been asked in your response have been really helpful. First of all, I think I understand like the motivation behind this study pretty clearly now. Like the questions that have been raised with regard to RGE, issues with direct and indirect effects underlying the PGS, like those issues are there and you're not trying to fix those problems. So I understand it. And I also understand that while we have possibly developed solutions to better modeling the G component, maybe even the E component. We've developed better ways of measuring the outcome component. We really haven't really thought that deeply about how best to interpret the B sub 3 component, which is what you're doing here, I think, is to help us better understand, like, what would it mean if you find a significant G by E effect? Is it really a G by E effect, or is it more just an over effect of the environment, as you've been saying? Well, the question I wanted to ask was really just more like an applied level type question. So what would you say should be the best practices then for researchers who are interested in testing G by E? So you've reviewed so many papers now that have just, you know, produced these G by E findings with PGSs, and they may not be interpreting them exactly correctly when you have such a large main effect of the E. Would you say that we should, as best practices, test a vanilla model, but also test for heteroscedasticity as well, and to determine whether or not there is this sort of scaling G by E underlying it? I just wanted to hear your thoughts about that.
That's a great question. You know, surely to drive up the citation counts of this paper, I would definitely suggest using this model. But I think the more honest answer would be, and this is terrain we tackled a little bit in the sociological science paper, but one thing would be a real emphasis on scrutinizing the nature of the outcome variable and you know, really asking about its psychometric properties. Is it, you know, does it have a ceiling or floor? Are we, if it's ordinal, has that been accounted for appropriately? This heteroscedasticity bit is one. If you have skew, has that been addressed? I think those are all important. You can look at, in our paper, you know, the skew thing definitely has some implications for findings, right? So that's a big one that people just typically don't even bother with. Asking about the measurement error of the environment is, I think, a good one. Here we have something measured with very high fidelity. Like, I would be okay calling birth year almost perfectly measured, at least in the sense that I don't think people are kind of writing down the wrong birth year, but that's often not the case. Sam and I have thought about scenarios wherein you could ask, you know, we oftentimes have people going straight to fairly specific environmental variables that they care about. And I think often you could potentially do more agnostic things that asked about variation in association across environments prior to stating specifically what you thought that environment might be. That might be useful. I guess, you know, the incentive is just to get a B3 coefficient that's significant and to call it done. And I just, you know, fundamentally want that to be not the kind of thing people are shooting for. And so there are a lot of different ways one could kind of add complexity to that, but the nature of the outcome, whether you have the right E and how it's measured, those to me seem like pretty big issues that are under addressed at present.
Thanks.
Ben and Sam, I think I have a more general question that I think Q's is a special case of, but I'd like your thoughts. Going back to this birth year thing for BMI, I'm wondering if you think that there's just a major model misspecification issue in terms of the environment between like 1925 and 1960 being a linear environmental term, and whether adding some different versions of what E means would actually fix this problem. And I say that Q is a special case is that I interpreted what Q was saying as a mismeasurement of G that's correlated with the environment when he were talking about like the two sets of SNPs. But if you use G throughout, then you're kind of like differentially mismeasuring G in some environments, but not others. So I think those both fit under a classification of just model misspecification. And I think right now the model acts as if it's specified correctly and then asks about heteroscedasticity. Is that how much of that is correct and how much of it would you like to add on to?
So I think I missed that last part. So the last part is, say the last question you did?
This is maybe it's the same kind of question I had before in some sense, but asking whether the model that you're building in the code has a kind of a baseline assumption that the model is specified correctly and then moves from there, rather than asking like, how does the model perform if I have some, at least some problems on the measurement side of E or the measurement side of G or so on.
Okay, so let me answer that, and then I'll come back to the first one. So what I would say is that our approach basically, we're assuming that this model is sufficient. This is a fairly flexible model, but yeah, you could certainly imagine deviations from that assumption that might be concerning, right? So maybe mismeasurement of E is one, but I would just again emphasize that this model is at least allowing for features that might be relevant that aren't allowed for by the vanilla model, specifically heteroscedasticity. So I think it's an improvement, even if it's not a kind of cure-all for the various issues we've discussed. Now, with respect to your other question about whether the BMI effect should be treated as linear, I think that's an interesting question. My concern would just be that we don't have sufficient data in the HRS probably to do a ton better. I'm thinking specifically about like this kind of observation where, I don't know, I'm not seeing huge nonlinearities in the quantiles at least. I haven't examined this thoroughly, so it might be that there's something I missed. But I think at least at a first pass, I'm okay with the linear assumption in the HRS data. Whether it's the correct assumption, full stop, I don't know. That's an interesting question. I'm surprised. In the course of writing this, I've tried to find good epidemiological evidence about this, and the stuff I've found is pretty descriptive. It often looks like this kind of thing. And that seems like an interesting question that we potentially answer with the in-Haynes data, but that I don't think people have looked at in a fairly precise way yet. So that's a good point.
Jason, I think I'm really sympathetic to your desire to, maybe be able to do more flexible things with sort of the model we specified. Because in a lot of ways, we're just taking this base, beta 1, beta 2, beta 3, 3 is the interaction, and then rolling with that in a very particular way with heteroscedasticity. Yeah, I mean, I think we are still thinking about how to add some of the elements you're talking about in it. The model that we're fitting is more complicated than just fitting that linear model, because you're having an interaction term with this latent variable that is the sort of error term, which normally you don't have to estimate. So I mean, it's just a little more complicated. We've added a complication, and I think layering on more complications, just they kind of interact. I guess that's there's interactive complications. But I mean, I think all the things you're saying are true.
This does not speak directly to the BMI linearity issue, but I would note that Paige Harden and I wrote a paper wherein we were looking at an interaction that we started modeling with the cumulative link logit model, and we got worried about whether that was appropriate for a variety of reasons, one of them being the linearity assumption. And we did try to explore a bunch of, I think we ended up with kind of several competing models in the paper that all were pointing in the same basic direction, including using something like Lois to very flexibly model bits and pieces of that. So I think that paper could be at least one example of how you could expand the horizon in terms of looking at a broader class of models that might help you better understand whether the linearity assumption is really crucial or correct, or in fact insufficient or something like that.
Maybe a different way of asking is essentially having another set of simulations assessing how bad linearity or how bad the model would have to be to produce these results, where it's actually not heteroscedasticity in some sense, that it's actually because I think of Q's toy example is fitting here, which is that I interpret what he said as you've got a case where you're systematically mismeasuring G based on the environment that's correlated with the environment. And I agree. I mean, I agree you can't do all the cases. I mean, I would still elevate RGE as being more important for me. But when you have a linear assumption of a birth year that spans 40 years or something, I start to wonder how how good a approximation that is.
Yeah, no, that's a fair observation.
I have a question, Ben, too, just going back to thinking about VQTL effects and VPGS. Would you call, or would you think of a VPGS by E interaction as being evidence of a scaling effect, of scaling G by E?
Not necessarily. It's clearly, it's definitely related. You know, if you have kind of genetic variation being associated with phenotypic variation that way, it's definitely kind of echoing the scaling thing. But there might be more to it, I guess. So it would definitely, I think, be one reason you could end up with scaling G by E, or scaling in this case, I guess, G by G. But there might be more to it than that. I don't think it would be that simple.
Jason, I think there's something interesting that you're kind of talking about, like with the measure of G across time. And I mean, I'd have to think about this more, but I think there's something weird about these, like when you're using a polygenic score, and so like I interpreted what you were saying by the mismeasure of G over time is that the idea that like the true latent polygenic score that would capture the additive genetic effects is different in one time period than another, and you know, you're using the same one across time. And you're even using a third one probably that's like neither doesn't map onto either like the post or like the later time period or the earlier time period. But I also, I mean, we talked about this a little bit in the sociological science paper, but I think that there's this big issue of like even I think that you can use polygenic scores that kind of detect to some extent with a lot of, you know, the caveats we're talking about here to detect like the existence of gene environment interactions. But the sign is kind of weird because it can hinge on like if the true polygenic score is different in one time than another, I mean, that is evidence of gene environment interaction, but it's not like the beta coefficient is actually going to align not with like when the penetrance is in the abstract bigger, but it's just like which score it maps onto better. Like where there's less measurement error between the score you're using and then the true score in that period, which is sort of, I think, counterintuitive. Like you might get a negative G by E effect. Like you might learn that like later. So all of this is say you ran a GWAS on the genetics for BMI change over time, and you run a GWAS on all sort of like people born in later birth cohorts. And over time, there is increasing genetic penetrance, but you do this interaction term. I think you would actually get, like, you would get a G by E saying that, like, I mean, the point is that you could get a G by E result that was positive or negative as a function of when you ran the GWAS, not when sort of what the true penetrance changes are over time. Which I mean, I don't know, there's just something weird about the idea of there being, like, I don't know if in the abstract you want to be measuring G perfectly over time. So I don't think you don't actually want a polygenic score that's different for every birth cohort. I don't think in these models, but also by using a single one, you're introducing this new complication that it's hard to interpret the coefficient. So I don't know, I think I need to think about this more, but I think it's an interesting point.
Yeah, that's a great point. It seems to me that assuming that the effect size or the penetrance of the PGS increases proportionally as the total variability increases, right? It's as if you're assuming that there's a scaling factor that's applied to all genetic variants in the genome. So all SNPs effect size increases proportionally. So that's sort of the underlying assumption here. So that's why I struggle to understand that whole example I constructed. In my case, a set of genes have changed the effect. Not a set of genes has the same effect. So it doesn't necessarily fit into the, the framework you built here. I think that's the key difference here.
Yeah. I mean, I guess one thing that this is all making me kind of appreciate and realize is that the scope of possibilities that are out there along the lines of what Q and Lauren and Jason and James have asked about are so much broader than what are analyzed in practice. And so to the extent that more of these things were to filter into the literature, that would, I think, be great.
Yeah, so I guess Ben just presented on one thing, one reason why you might get a significant beta 3, which is probably a little different than you would expect, or is a little different than just like this complicated, genes matter differently in different contexts interpretation. But there's I don't know, there's probably 10 other reasons you get a significant beta 3 that don't map onto that as well. And I don't know. I think, yeah, as a field, we probably need to flush those out as a way of like, you know, I think the robust research is going to get more credit as if there's sort of, you know, not a sort of alongside it a bunch of like less robust research.
I have to step away for just one moment. Excuse me.
Sam, while he's away, you can. We'll need to wrap up anyway. But one thing is whether you think there's a large, like how large the class is, how big is the worry. Like I think birth year is a great toy example that you all picked. I struggle to find like the fourth and fifth example that I'd be really worried about, because I mean, birth year has these classic features that you just like everyone nods that, yeah, BMI probably is going to be this good one for birth year. But I'm wondering not necessarily the second example, but maybe the fourth or fifth.
Yeah, I mean, what's interesting though is that I mean, if we're just sort of going back to this pad example of Ben getting a bunch of these papers with just sort of gene-environment interactions, I mean, I think birth year and socioeconomic status probably are like two that I think probably have this issue that they like sort of they do have sort of distributional differences across the various levels. And I think like it might be that like the GxE with that fifth candidate isn't actually that common. I mean, it could be that just even if it was just those two, that would encompass a lot of these like beta 1, beta 2, beta 3 models. But you know, I don't know for sure. But I think that's a fun endeavor. Ben, can you think of like good candidate dimmers besides sort of birth year and SES?
Oh, well, I think another way of saying it would be that like often the outcomes are going to be such that you're going to end up with the dimmer effect, not so much because it has to be that way, but because we've not measured the outcome with very high fidelity. So if you have a ceiling or a floor on your outcome, we're only going to get variation where we can, right? And so the ceiling or floor, you know, so long as there's a main effect of the environment on the outcome and it's true metric, the ceiling and the floor are going to be such that we end up with restricted amounts of variation as a function of the environment, even if that were not the case in kind of true outcome metric. So yeah, my inclination is to think that this stuff is likely to be rampant, even if it's often kind of being caused by things we could potentially address. Well, things that you might not think were immediately due to it being a dimmer in some sense, but that would effectively manifest as it being a dimmer.
Yeah, I was thinking, I remember a few years ago when you came to Ann Arbor, we were talking about that paper I've been working on with increasing education across cohorts and in particular high school graduation. And I think that's a really great example of what you're saying, these variables with a floor and a ceiling. And I find this really, I don't call it a GxE effect, but a really strong interaction if you interact kind of changing high school graduation over time with the education polygenic score. And I think it is more because we've just, high school graduation rates have increased so, enormously across cohorts. And so it's really, we're reaching this kind of ceiling effect that is kind of, leading to this kind of GxE interaction effect. Because you almost can't just get any more high school education, and but it's also a function of birth cohort, like in everything else, and SES, but that was an example that came to mind.
Yeah.
Feeling from Jason, I think age is a good one too. Like independent of birth cohort, I think we would see variation in a lot of variant shift in a lot of outcome variables across age.
Height.
This is just a fun game.
In the simulations I've been doing recently, I've been increasingly concerned about ceilings and floors. They just they ruin things. And if you think about how we often measure depression or any number of things, we effectively only measure at one end of the spectrum, and that's a huge problem. And so that's actually something that if you look at that social science paper I linked to, you can end up with, I think, a version of what we have in Figure 1 there, wherein changing levels of prevalence of something can be easily misunderstood as GxE if you're not very careful, purely because if something is basically not prevalent in one environment, you're going to think about it as being a very flat association. But if that is only due to the fact that there's an intercept shift that put everybody above the liability threshold, you could really kind of misunderstand what's going on. Yeah, that's, I think, a kind of related example where mismeasurement of the outcome is leading to spurious G/E findings.
And I have a quick last question that I mean, because we haven't talked about this, but like, you know, in some sense, you presented a paper that's all about like how, you know, a lot of times G/E findings are a little different than you expect, and it's like harder to detect G/E, which I think is true. Like, it is hard to detect G/E. I think my prior though isn't that like there's no G/E in the world. Like people are detecting all this stuff and it's just noise. Like, I kind of believe that my prior is that there is sort of rampant G/E, but it's just really hard to sift out, you know, from this, like, you know, gene environment correlation, from these floor ceiling effects, from like the SES per theory sort of heteroscedasticity. What do you think about it?
Yeah, I agree with that. Like, I don't have any stance on whether there should or shouldn't be G/E, or it's more just like, I think it's harder to detect, and it's harder to detect the thing we kind of have conventionally convinced ourselves that we care about. And so I think we should work on that, expand the kind of range of models we're willing to kind of think about that would drive our data. But yeah, it's not that I think there's no G/E. It's just that for some reason we really incentivize the identification of kind of G/E, and I think that's like a slightly absurd game that we've stuck ourselves with.
Yeah, I also sort of think like not all G/E is equal. Like I'm pretty convinced that genetic effects tend to be contingent on a lot of things, like including, you know, various environmental factors. Like, maybe the world isn't, but like for me, I want to know, like, why does this G/E particularly interesting? Like, I mean, I think, Jason, you're like smoking and taxes G/E is like the classic example of like that clearly matters, right? Like, we want to know that as a policymaker, or as just a human living in this world.
Or I think, you know, Robbie's paper where he's showing a changing levels of pleiotropy between the educational attainment SNPs and the smoking SNPs. Like, to me, that's like an interesting, like, I don't even know if you want to call it G/E. That's like not necessarily the relevant kind of term, but like that's an interesting conceptual model for something that's changing in the world that's interesting.
Ben, I know we're at time, and thanks for such a great conversation and presentation. I know you have some more meetings, including with me, so we'll move on to that and say goodbye to everybody, and thanks everyone for coming.
Thanks all.